Drug-resistant pathogenic microbes are a growing risk, and we need new methods for controlling them. With me to discuss possible strategies, how to develop self-assembled antimicrobial materials for future biomedical application, is Dr. Jennifer Payne from Monash University. Dr. Payne, thank you so much for taking the time out of your busy schedule.
No, thank you for having me, Alistair. It's great to be here, and welcome to you from Switzerland, and I'm joining you from Australia.
Thank you. Dr. Payne, your recent research shows how a certain bacteria can get caught in a net of self-assembling proteins that have vancomycin attached, thus locking the bacterium in place and destroying it. Can you first tell us more about the background of that research?
Sure. So our own body uses a very similar mechanism to kill bacteria, and that's called netosis, or neutrophil extracellular traps. And neutrophils are one of our immune cells that are part of our innate immune system, and think of them kind of like our first responders. They're patrollers searching out for potential invaders in our body. And it's not just neutrophils that do this, but also macrophages, and cells are part of our innate immune system, can form these net-like structures. So what is a net? A net is exactly what you think of, like a fishing net, that sort of thing, but think of it in miniature. And we're talking about a cell in your body producing a net, and this net is actually their DNA. So they eject out DNA. One of the processes basically by suicide and ejecting out their DNA, putting it over the top of the bacteria and entrapping it that way. And this DNA is actually embedded with a range of different molecules, ranging from ones that lyse the bacteria, act as antimicrobials. So not just straight DNA is in there. So it acts like a net, but also a deadly net that's embedded with these killing mechanisms already in there. So using that as kind of an inspiration, we are having a conversation around the water cooler, as you call it, kind of thing, with the lab next door. And our lab works on antimicrobials, antibiotics, drugs that basically kill bacteria, and also understanding the innate immune system and how antibiotics interplay with superbugs and how that affects our immune system as well. So that three-way interplay. While the lab next door works on peptides that self-assemble. And so they create fibrous structures that are able to self-assemble and more of the biomaterials side of things, while we were coming at it from a superbug innate immune system angle. And speaking about their nanofibers, they're like, oh, we construct these self-assembly, they build themselves. So you just put in a single monomer that's really tiny, and then they can build themselves up and create really long nanofibrous chains. And we're like, oh, that's really cool and amazing. And like, have you tried sticking anything onto them? You're like, no. Well, we've got this antibiotic that has a group on it that allows us to do some simple quick chemistry to attach it on to different things. Can we easily incorporate the corresponding group onto one of your monomers to be able to incorporate it into a growing beta fibril? And they're like, yeah, that's feasible. Do you have any idea how big of a group we can stick onto it? No. We'll just suck it and see how big we can go and what we can put on. And lo and behold, put it on, and we're getting, we're like, oh, it's probably just going to dismantle the fiber structure and we won't get any fibers forming. It won't work. And if we just have the vancomycin attached onto the monomer, yeah, it didn't work. Okay, yeah, sure. But what happens if we mix in a bit more of the monomer and like give it a bit more spacing? Does that then work? And what we got was these fibrous structures starting to form when we mixed the monomer that's attached onto vancomycin plus the single monomers. So we're actually getting fibers. And so that was like the first step of, wow, we can actually do this, to then going, all right, well, now we've got fibers forming. This is very much similar to the net process that I just told you about that happens in us. So does this then have any activity? Can we then use this to control superbugs? So we're like, okay, well, we'll throw in some superbugs and see what happens. And we'll go, okay, well, the main one that's a real major problem is MRSA, or methicillin-resistant Staph aureus. Let's see what happens when we mix these guys together and see what happens. And what we got was actual activity against them. So we were really actually surprised that we could get fibers forming that were embedding this vancomycin in the first place, let alone that we then had activity. So it was all a serendipitous kind of occasion of let's suck it and see and have some fun as scientists combining two labs experiences together to create something that mimics our own immune system. So it was really inspirational in that regard. Some fun science.
And is there an advantage of using a net of proteins without an antibiotic and only introducing this bacterium after the bacterium has been locked in place? And what do you think are some advantages or disadvantages of both methods?
Mm-hmm. So when we were looking at these fibers that formed when there was no vancomycin, no antibiotic present, we weren't really seeing the wrapping effect. So one of my favorite photos looks like in confocal and doing a 3D reconstruction of what the fibers look like with the bacteria, is that the fibers have grown up and around, and then there's this Staph aureus sitting here, and then this fiber formation, and it kind of looks like a goblet of fire. So because the fibers are in red, so we've got this glowing mass of fibers down below, and then lo and behold, the Staph aureus held up in this cup of fibers, which was pretty cool. It looked really neat under the microscope. But we don't see that when we've just got the fibers there by themselves. So if they don't have the vancomycin monomer in there, we're not getting that attachment and locking on. The bacteria are kind of there, but they can kind of get washed out as well. So we're not getting that entrapment into the fiber. So it actually needs something in that fiber forming that binds onto the bacteria to get that full entrapment happening. So to bring that back to your question, the advantage of using this net that has the embedded antimicrobials in it is that we're actually getting binding onto the bacteria and stopping them that way, mimicking very much exactly that DNA net that happens with our immune system. That's not just straight DNA, has those embedded antimicrobials, so they do that same sort of thing. And we don't think you would get the same sort of bacterial control if we just had the straight protein fibers there. That you would get a growing out, and the bacteria actually kind of liking that environment to grow into. So it needs that added antimicrobial there to stop the growth and entrap the bacteria. Yeah.
And would a similar strategy work with other antibiotics? Or in your opinion, why would it not work?
So good question. Vancomycin, we need to go into a little bit about how vancomycin works as an antibiotic. So it is an antibiotic that binds to the cell surface to kill bacteria. And not all antibiotics work this way. So some come into the cell and bind and stop intracellular processes. So I don't think this strategy would work with an antibiotic that requires getting into the cell to function. Because if it's embedded in a fiber, it just can't get down into the site where it needs to function. So let's rule out those ones, though I haven't tested that, so I can't say that for certain. That's just theory that I would say that they wouldn't function. But we then have a suite of different antimicrobials that bind to the surface of bacteria and fungi that we could potentially then use in the same sort of strategy. Or we mix and match and add in ones that could then be potentially released off from the fiber that act intracellularly versus ones that bind to the surface, which mimics more of that net situation, because it's not just one mechanism of action that's present in a human net. So it could mimic that mixed antimicrobial present rather than just the straight vancomycin that we're talking about in this paper. So vancomycin works by binding to the surface, and in particular to diala, which is part of lipid 2, which is an essential cell wall precursor. And I said precursor, so it's not actually it ends up forming part of the major structure of the cell wall. But when it's a precursor, it's down really close to the plasma membrane. So if you think about that, you've got plasma membrane, cell wall, which means that you need vancomycin to get through the cell wall down to the closer to the plasma membrane to function. And if you've got vancomycin stuck onto a big long fiber, how is that getting down and through the wall to get down to the septum to actually function? Which is one of the questions that's really remaining from this paper of like, how is this actually working as an antimicrobial if vancomycin's stuck onto the fiber and can't get down to the membrane where it normally works? So it's probably still binding to the cell wall because there's, I guess, DLR still throughout the wall, but just not where vancomycin needs to act down at the membrane. So it's probably binding onto that up towards the surface further. Or maybe there's smaller monomers that are released from the growing fiber and that's getting down, or maybe it's just binding to the surface of the bacteria and that's then slowing down growth because it's getting wrapped up in it, physically strained. So how this actually works and its mechanism of action is really something that we need to dig into more to be able to kind of answer that question of what other antimicrobials would work with a direct interchange with vancomycin. But definitely a potential there of being able to mix and match and add in different activities into this fiber structure so that it's not just one activity and mobilized onto the net structure. So potential.
And talking a bit more about the advantages of this immobilization and antibiotic tactic over using only antibiotics, what do you think are the key advantages, and would those outweigh also added costs?
Yeah, so immobilizing has the advantage that you're then keeping the antibiotic where it's needed. So if we were to create this self-assembled fiber network in, say, like a diabetic wound or a chronic wound type situation, you then have the antibiotic there where it's needed, rather than, say, a systemic antibiotic given. So something where you've either taken the antibiotic orally or had it injected into your bloodstream. In those cases, the antibiotic can have off-target effects, so causing problems with the kidney and that sort of thing. And these are often why you can't give antibiotics at higher doses and why superbug infections become more problematic, is because you can't give those higher doses required to treat those infections. So if we can localize the antibiotic directly where it's needed, there's that potential to be able to kill the bacteria just where they are, rather than applying the drug directly to the whole body. And hopefully, fingers crossed, that then avoids those off-target effects. So that kind of potentially advantage hopefully outweighs some of the added costs. But if you're thinking in terms of chronic wounds type situation, clearing infections in those areas to be able to heal that wound. So I mentioned that we can add in vancomycin into that fiber, but why can't we just add in something that will then help that wound heal faster? So these fibers have been shown that they're great at forming a scaffold for cells, human cells, to grow into. So we know that you can put this fibrous gel into a wound and therefore our cells could then grow into this. So it could form a mesh that would allow our body to heal and form the necessary structures across, keeping the infection at bay. But why not add in some growth factors or VEGF or that kind of thing to help the vasculature form and the cells migrate into that area so that you get a faster wound healing happening through this gel. So that would then help speed up that chronic wound healing by combining it not just with a direct antibiotic, but creating something more of a complete healing system rather than just a bacterial killing gel. Yeah.
Dr. Payne, let me wish you all the very best for your further research. There are big challenges still to solve. Good luck and thank you.
Thank you. Thank you yourself.